So now we have on our agenda our first speaker, who is Tom Lin. He's an instructor from the Washington University in St. Louis. Today, Tom's presentation is on top-down characterization of post-translational arginylation using the electron activated dissociation, or EAD. Today's presentation, you're going to learn about the top-down proteomics workflow for the characterization and quantification of the calreticulin arginylation. And we're going to hear about the MS1 profile, MS2 fragmentation, where that was characterized with the EAD fragmentation. Tom, I will hand this over to you now for hearing about your exciting research that you're doing with the xenotoph.
Thank you, Tatiana, for the introduction, and really my pleasure to give this talk. Yeah. Good morning, everyone. It's my great pleasure to give this talk on arginylation using the top-down proteomics. A little bit background on the arginylation. So arginylation, arginine is as an amino acid residue is charged onto the tRNA in the middle of the slide, and this intermediate can be used for translation by the ribosome on the top right, and also can be used for arginylation by the enzyme called arginine transferase 1, ATE1, to install the N-terminal arginylation and the side chain arginylation on the amino acid. Arginylation is an essential PTM, but a hard one to discover by proteomics. And the ATE1 knockout in the conditional knockout mouse models will show that the mice after birth will die in 1 week due to the phenotypes of shallow and rapid breathing, and eventually resulting in the accumulation of the air in the abdominal cavity, which will result into the death. So the main challenge is to differentiate the post-translational arginylation from a peptide sequence from the translational arginylation, from the translational arginine. As you can see from the top right, if you search arginine as a PTM, you will get the top sequence with the N-terminal arginylation. But if you search the normal proteomics, we allowing for the miscleavages, and then you will get the R residue on the left side as a trypsin miscleavage. So it's really hard to differentiate whether this R is from the post-translational modification or it's a miscleavage. Another example shows that one MS2 spectra can be assigned to different peptide sequence based on your search strategies. And this is the proteomics bias that no one wants. So over the past 3 years, I have established an unbiased workflow to discover arginylation sites from the ATE1 assay. Using ATE1 assay, we use ATE1 to install the arginine 10 and arginine 0 separately to the same proteome from the cell lysate, and then we mix them. The mixed proteome will go through the normal proteomics prep until the digested tryptic peptides. Because the arginated peptides in the peptides is so low abundance, we have to use the high pH fractionation to get the peptides in around 30 fractions. And then we inject each of the fraction to the LC mass for the analysis. Using this way, we can search for all the peptide IDs, and then from there, we only look for the paired peptides modified by the heavy and light arginine. And then we look into their MS1 pairs for the signatures of a 10 Dalton mass shift. Using all the MS1 paired MS1 scans, we can calculate the heavy letter ratios for our further inspection to identify the final list of the sites. There are three advantages here. One is that we can eliminate the bias from the ribosome synthesis when we start from the cell lysis, not from the lab cells. And two is that the workflow can be used for any proteomes, so we are not limited for any sample types. And three is that the arginine 10 signal from the MS1 and MS2 is purely dependent on the ATE1, so it's post-translational. And one pitfall of this ex vivo workflow is that the sites reviewed from here do not necessarily mean that they are also happening in the cells or in vivo. To facilitate the data analysis, we have developed a software called Arginylation plot. The design and the flowchart of the software is shown up here. So basically, from the raw data, we will do two steps. Step one is to convert them into the MZ XML file as a general format for software recognition. And then we search for the results and the peptide IDs. If we find the MS2 pair, then the software will go to the MZ XML file to extract all the paired MS1 scans. All the MS1 scans will be summarized into the heavy letter ratio, and the relevant MS2 scans will also be exported for the database index. We have been able to discover over 200 arginylation sites from 1,337 pairs of paired peptides. And these arginylation sites were discovered from 14 samples, including the iPSC cardiofibroblast iPSC cells and the cardiomyocyte cells. We also applied the same platform into the Hectorine 3T and other 5 cancer cell lines. And moving one more step forward, we applied this platform to the patient samples for example, the patients with the heart failure, the brain, and also other brains with cognitive dysfunctions such as Parkinson's disease with dementia, and then also Alzheimer's disease. So if you look at the heavy letter ratio from the MS1 scans, the ratios of the 1,000 over 1,000 pairs is centered perfectly at the heavy letter ratio 1.0, which is a hypothetical ratio from the design of our workflow. Here is a table of all 229 sites ranked based on their numbers of detections. The sites are categorized into 4 types of arginylation based on the modification masses, and R as the arginylation, RO3 as arginylation after cysteine trioxidation, RO2 as arginylation after cysteine dioxidation, and RDME as arginylation after the deamidation of the asparagine and the glutamine. To validate some of the sites, we use the standard peptides on the top to compare their chromatographic MS1 and MS2 behaviors of 7 sites. The middle of the figure shows a representative MS2 spectra containing the arginine 10 and arginine 0 modifications. In addition, on the bottom left, we used an in-bacteria arginylation assay to confirm 2 sets, ER01A E24 and SSBP E17. Using the Y-type and AT1 knockout cells, we show that arginylation in calreticulin ER01A and SSBP is ATE1 dependent. And when we overexpress the ATE1 in the knockout cells on the bottom right, we show that arginylation level is dose dependent on the AT1 expression levels. Since this is the first unbiased platform for arginylation discovery, we have built a public database for everyone to review and download our data. Arginylation sites were indexed from the peptide mixture protein, human proteome, and mouse proteome samples. And everyone is encouraged to visit our website and give us any feedbacks. Among all the proteins and sites, calreticulin is the top one high frequent target from our bottom-up data, and the calreticulin has also been confirmed for its arginylation using the pull-down experiments as shown up on the top. And in addition, we also purified the calreticulin protein with the E18 open site, and this has been confirmed for its arginylation in vitro using the AT1 assay. Let's take a closer look at the calreticulin. Calreticulin is a highly conserved chaperone protein which resides primarily in the endoplasmic reticulum, the ER, and it's involved in a variety of cellular processes, including cell adhesion, ER trafficking, protein folding, and calcium homeostasis. Except for its biological importance, we also have other scientific motivations for characterizing the calreticulin arginylation using top-down proteomics. First, the bottom-up proteomics always struggles to identify the endogenous arginylation. Secondly, the bottom proteomics is usually unable to quantify the proteoforms since the bottom-up only look at the peptide level identification and quantification. And the top-down proteomics has never been used to characterize the low abundance arginylation. As mentioned, calreticulin is kind of the positive control in the arginylation field, and it's a high frequent target in our bottom-up data. So to establish the top-down workflow, we have constructed a cell pull-down and the purification process to obtain the calreticulin and the arginated calreticulin, CALR. In the cells, the signaling peptide amino acid 1,2,17 is cleaved after the protein synthesis. Taking advantage of this process, we have inserted an arginine residue before the signaling peptide as shown on the bottom left. And using this way, the cells will cleave the signaling peptide after the transfection, and then produce the Y tab and the arginated calreticulin. The halo tag in the calreticulin in the middle will be cleaved after with the tab protease to produce the Y tab calreticulin and arginated calreticulin. Here is a detailed workflow to show how we prepared our samples. The unmodified calreticulin in lane 1 is prepared by overexpressing the plasmid in the AT1 knockout cells. The fully arginated calreticulin in lane 5 was prepared similarly with the unmodified calreticulin in lane 1. Lane 2, 3, and 4 are the endogenously arginated calreticulin, where we co-transfected AT1 with the Y-tab calreticulin plasmid. In this way, the AT1 is able to act as the enzyme to arginate the overexpressed Y-tab calreticulin. So from this experiment, we can obtain 3 types of samples, right? The sample with 0% arginylation in the lane 1, the sample with endogenous level arginylation in the lane 2, 3, and 4, and then the calreticulin with a high arginylation in the lane 5. We used Waters M-class UPLC for the chromatography and the SciX Zenitov 7600 system for the mass spectrometry. And the right side of the slide shows the LC gradient using a 20-minute method and a C4 column due to the fact that calreticulin is large with a molecular weight of 50 kDa. Using this method, we were able to get consistent retentions and get great peak shapes from the chromatography. The top two panels shows the total M chromatogram of the calreticulin and the arginated calreticulin samples with the presence of the TAV protease as the together with the calreticulin as the two obvious species. The bottom panel shows the extracted M chromatogram of the calreticulin and the arginated calreticulin at different M/Z values. Looking into their MS1, both protein forms showed the highest intensity at charge 58. If you look at the right as a zoom-in version of the individual charge state at 58, you can clearly see that the 156 Dalton mass shift caused by the arginylation in the bottom. We then sent the two species for the EAD fragmentation and get around 10 and 11 sequence coverage for both protein forms. Because arginylation happens mostly on the protein N-terminals, and the EAD shows efficient fragmentation on the N-terminal, so the missed sequence coverage did not affect our arginylation studies in this analysis. Here is a annotated MS2 spectra for each calreticulin species, the wild-type calreticulin on the left and the arginated calreticulin on the right. As you can see that the arginylation on the N-terminus did boost the intensities of the N-terminal C series of ions significantly in the arginated calreticulin due to the addition of the R residue, which is highly ionizable. We did try to find the reasons why calreticulin had such low sequence coverage, especially for the middle of the sequence. There are two reasons. One is that calreticulin is a big protein, as mentioned before. Getting it ionized and fragmented well is challenging in general. Secondly, the calcium binding domain has 114 aspartate and glutamate residues, which may further reduce its ionization and fragmentation efficiencies, as shown in this figure. So after this, we went on to test if our top-down proteomics can be used for arginylation quantification. The first experiments that we did was to mix the wild-type and arginated calreticulin artificially at different ratios. As can be seen from the left side, you can clearly see the increase of the CALR signal and the decrease of the arginated calreticulin signal from the mixed samples. Using the MS1 signals, we show that the quantification of the two species correlates nicely with their mixed ratios. We then try to identify and quantify the arginylation and endogenous level using sample 2 and 4. By just looking at the MS1 spectra, we could not confidently identify the arginylation due to the low signal. However, when sending both species to the MS2 for fragmentation, we can clearly identify the N-terminal C series of ions containing the arginylation in sample 2, 3, and 4. Here, we use the sample 1 as a negative control and sample 5 as a positive control. Here is a representative zoom-in figure for the C3-ans with arginylation in all 5 samples. Using this way, we can confidently identify the arginylation using MS2 at the endogenous levels. When we then went back to the MS1 at multiple charge states and calculated the arginylation levels, and the percentage of the arginylation of each sample is displayed in the figure B on the bottom side of this slide. Yeah. One thing that I want to point out is that the arginylation level in sample 5 is supposed to be 100%. However, it is only showing 96%. We looked into the MS1 spectra and clearly saw the unmodified form of the calreticulin, which showed us at almost every charge state. The bottom right figure is showing a zoom-in version of the 3 charge state, indicating the presence of the unarginated calreticulin. So this is a clear evidence that the de-arginylation might have happened in the cells, and this is a direction that no one has investigated before, and our group is very interested in the de-arginylation field. Moving forward from the endogenous arginylation, we also applied our platform to the in vitro arginylation system, similar to our bottom-up workflow. Using a commercial calreticulin and ATE1 assay, we can arginylate the calreticulin and monitor the arginylated product using the top-down proteomics workflow here. Similarly, we have established a unbiased arginylation assay as shown in the top left corner. Briefly, calreticulin on halo beads can be reconstituted into the ATE1 assay, and the assay components were then washed away, and TAV protease was added to cleave the calreticulin proteins. And we did the isotopic arginylation using this new assay, and can see the calreticulin bands after the cleavage on the silver staining. Using the top-down, we can see the arginylation species modified by both arginine 0 on the top and arginine 10 on the bottom. The relative arginylation efficiency was calculated as 63% and 73.8%. MS2 fragments on the right side clearly shows the C series of ans with a mass difference of 10 Dalton differences. The sequence coverage of the isotopic modified arginine proteoforms is shown on this slide. Beyond the top-down calculation of the arginylation, we also applied our top-down workflow to the arginylation studies of other proteins. Here we show the arginylation species of the ER01A and SSBP generated from the in-bacteria arginylation assay. You can see on the right side in the middle, the plus R basically means the arginated species. And you can also observe 2 other species. One is the unmodified species on the left side, marked as wet type. And you can also see a 3rd species, which is bacteria specific. So this is a lucinated arginated species, lucinated and arginated species, as shown up in the sequence as a unique product produced by the bacteria enzyme called LFTR, lucylphenylalanine transferase. So to To summarize, I hope that you are convinced that we can use bottom-up proteomics to unbiasedly profile the arginylation from different proteomes. And using the calreticulin and xenotop 7600 system, we established the first top-down approach for the characterization of the arginylation. And the EAD is efficient to characterize the N-terminal arginylation. The workflow can quantify endogenous arginylation using a combination of MS1 and MS2 features. This platform can be used for endogenous, in vitro, unbiased, and in bacteria AT1 assays. And the arginylation can be monitored using either single channel or isotopically, different channels of arginylation. So in the future, we would like to use the top-down identification to identify and quantify the endogenous and the in vitro arginylation in the unknown protein substrates. And also, we would like to use the isotopic arginylation to unbiasedly discover the protein substrates from the proteomes from different biological samples. And as mentioned a few minutes ago, that the mechanism of producing the de-arginated species is of our high interest. With that, I would like to thank my lab, especially my advisor Ben Garcia, and my lab members and my past lab members showing up here. I would like to thank our key collaborators, Yi Zhang from Case Western Reserve University, and also Anna Kashina from University of Pennsylvania, who is a pioneer in the arginylation field for the past 20-30 years. And also, I would like to thank my computational support, Chenfeng Zhao and Becky Han, to write the computational software for data analysis for both bottom-up and top-down data. At the end, I would like to thank my NCI R21 funding and my WSU BMB seed grant funding, and also I was supported as a REC scholar from the NIA Peace Study grant, and also several grants, several NIH and NSF grants from Ben Garcia. Yeah, I will end my talk here, and I really appreciate your time to listen to my talk, and I'm happy to answer any questions that you may have.